Hi, my name is Huaming Sun. I'm a PhD student in Shell Lab and Cooking Lab at Worcester Polytechnic Institute. In this video, I will talk about my research using machine learning to understand the messenger RNA degradation mechanism in mycobacteria. The reason we study mycobacteria is because one of the mycobacteria, Mycobacteria tuberculosis, is the causative agent of tuberculosis, which remains a global public health crisis in 2020. What's worse is that such situation hasn't been improved, and the total number of TB cases remain largely unchanged over the years. Generally, it will take over 6 months to treat TB. During this process, MTB inhabits a complex and stressful host environment. It encounters antibiotics within the context of multiple stressors, such as hypoxia and low pH. Still, it's able to survive and proliferate. Also, further developed antibiotic tolerance will make the treatment even harder and facilitate drug resistance. So all of this raises a very interesting question, which is what makes TB so successful in response to stress? So my research focused on one of the possible mechanisms, that is the post-transcriptional regulation, the messenger RNA degradation. Together with the transcription, messenger RNA degradation determines the messenger RNA abundance. By altering the messenger RNA abundance, messenger RNA degradation could contribute to stress response through 2 levels of regulation. One is the transcript-specific regulation, which could result in altered gene expression profiles. Another one is global regulation, which could contribute to the metabolic adaptation to energy-limited environments. And turns out this is exactly what happens when mycobacteria globally stabilize their messenger RNAs in response to severe energy stress. Also, previous research in MTB shows the half-life profiles had the variability both among genes and between conditions. Also, our preliminary result in MSMAG also shows there's a global stabilization when mycobacteria experience hypoxia condition. However, the messenger RNA features that control those variabilities are still not clear. So to identify those features is what I will focus on in this talk. In order to do that, we use machine learning to help us understand the mechanism by identifying the features that determine messenger RNA stability. After using rifampicin to block transcription initiation, we collect RNA over time for RNA-seq to quantify messenger RNA degradation transcriptome-wide using the non-pathogenic mycobacteria model MSMAG. After calculating half-life through the degradation profiles, we split genes into 4 different classes by their half-life. We then collect features from 5 different categories. Each one of them have features containing information about different parts of the messenger RNA. Now we have the data set containing all the genes in the mycobacteria, as well as their features to describe the characteristics of the genes. Then the random forest model will be trained to learn the association between those features and the half-life class, and will be further tested by making prediction of the half-life class given gene's feature. At the end, the model will rank the features by their ability to contribute to the prediction. Those more important features will have more distinct distribution over half-life classes, compared to those are less important. In mycobacteria, we are specifically interested in the leader and the leaderless genes, which are defined by if they have the 5' ATR. We'd like to see that if such difference could also indicate a different messenger RNA degradation mechanism. And also for each one of the gene type, we'd like to see that if there's would be any messenger RNA degradation mechanism difference between the log phase condition, which is the normal growing condition, and the hypoxia stress condition. So in order to do that, we started by developing machine learning model to predict half-life cases using features from only each one of those feature type, and evaluate their performance by calculating the F-score change relative to the random predicted model. And we find that for both leader and the leaderless genes, all of those models using individual feature type has the ability to predict the half-life. However, depending on the gene type and the condition, those model performance are very different, which indicate that the degradation mechanism could depend on the gene type and the condition. Then we try to develop model using all of the features from all different categories, which achieve among the top performance compared to the model using only individual feature type. However, the effect, as you can see, of each individual feature type does not seem to be additive in the combining model, which indicate that there might be a more complex nonlinear association among those features. To further confirm the difference between the leader and the leaderless genes, we trained a model using the same set of features for both type of genes under the log phase condition and hypoxia condition. Again, the result shows different performance in both of the condition for leader and the leaderless gene model. And for this talk, I will focus on the log phase and also the feature comparison between the leader and the leaderless gene model. And similar conclusion also found in the hypoxia condition. So first, by looking at the feature distribution over half-life cases, we can further explore how the feature value changes and the half-life changes. Here, we find that there are features are important to predict messenger RNA half-life for both leader and the leaderless genes. And those important features come from different categories. Here are 5 examples of those important features that from each one of the categories, as I mentioned earlier. And if we focus on the trend of change between those feature values and the half-life cas, we could see that those features really could work differently to control the messenger RNA stability, as they are changing towards different direction. And also, those features' relationship with the half-life cas could be nonlinear, as they are not always changing in a linearly way. Among those features, I'd like to focus on the nucleotide usage and the codon usage, which are 2 very close feature type. So because each individual codon contains 3 nucleotides, so it's entirely possible that the codon effects on the degradation we see here could just be driven by its nucleotide composition. However, as you can see here that, for example, the G frequency and the GGC codon frequency here, Not only that, the codon feature distribution does not entirely follow the distribution of its majority nucleotide distribution over the half-life Cas. But also, we find that given the exact same nucleotide composition, for example, here the CGG codon usage, we see that through the visualization, it actually have the very different relationship with the half-life. Such results indicate that the codon effect on the messenger RNA degradation may not be driven by its nucleotide composition. And further looking at those important features, we also find that some features are more important to predict half-life Cas for leader genes, while different features are more important for leaderless genes half-life prediction. So here are some examples of different codon usage effect on the leadered and the leaderless genes messenger RNA degradation. And we further look at the those codon usage and their relationship with the ribosome occupancy. And as you can see that they have very weak correlation, which indicate that the codon effect here may not be translation dependent. So the codon effect on the messenger RNA degradation could be both translation dependent and independent. As I mentioned earlier, the messenger RNA are globally stabilized in response to hypoxia stress. Our data also suggest that not only there's a global stabilization, but also such stabilizations are at different extent. As you can see, genes in log phase from different half-life categories are stabilized to a different extent under hypoxia condition. And this is true for both leadered and the leaderless genes. So such result makes us wondering that if we could also, using the feature to predict the extent of the messenger RNA degradation upon transition to hypoxia. And it turns out that we could. Although the model performed not as well as those models for each individual conditions for both leader and the leaderless genes, we're still able to see that the model's ability to predict such stabilization extent for both leader and the leaderless genes. And similarly, we can again look at the feature distribution over the half-life Cas, and we find that there are features important for both leader and the leaderless genes. And also there are features that are more important for one specific type of genes instead of both of them. So overall, our results suggest that we can develop machine learning model using the messenger RNA features to predict the messenger RNA degradation rate in log phase and hypoxia. We could also, using those features, to predict the extent of messenger RNA stabilization upon transition to hypoxia. And what's more is that we find the regulation of the messenger RNA degradation is actually achieved through not only one feature, but actually the combination of features from different categories. And the interaction among those features tend to be nonlinear, and they are not additive to in terms of the impact of the degradation. And also we find that the messenger RNA degradation determinants are actually different between the leader and the leaderless genes in mycobacteria. And they are also very different between the log phase and the hypoxia. So this is all for my talk. Thank you for your attention.